Okay, you guys, in this next video, which is actually the last video for the 4.2 notes, we're going to be looking at topic 2, the last topic in these notes. In topic 1, we talked about the cell cycle and basically the different things that take the different sequence of events that are going to take place in a typical cell that leads to cell division, and ultimately the cell will then divide and become two daughter cells. And then the cycle continues. And so in topic 2, though, we're going to now focus on how the cell cycle is being controlled in cells and in organisms, because it's very important that certain cells in your body who are not supposed to be dividing, that those cells are not dividing. And for certain cells that are supposed to be dividing, it's important that they are dividing and they're dividing correctly. And so there's a whole control system built into the cell cycle to help make sure that cells are dividing when they're supposed to and not dividing when they're not supposed to. And when they are dividing, making sure that they are dividing correctly and successfully producing beautiful daughter cells. And so that's what we're going to look at in topic 2, is how that takes place and some of these major components that help control that and make sure that's all working correctly. And so in this control system that is regulating the cell cycle, it basically is centered around these checkpoints that are embedded at different points in the cell cycle. And so a checkpoint is basically a moment in the cell cycle where the cell is going to pause and not continue moving on to the next part of the cell cycle until it verifies and makes sure that there are certain things happening within the cell. And then ultimately there's going to be these signals from inside and outside the cell that tell the cell, yeah, you're actually good to go. Go ahead and move on to the next part of the cell cycle. So in the cell cycle, there's actually 3 checkpoints built into it. There's the G1 checkpoint, which is kind of towards the end of the G1 phase after the cell has initially formed and grown and is getting ready to possibly start replicating its DNA in S phase and leading ultimately to cell division. And then there's the G2 checkpoint, which is at the very end of G2 before a cell is allowed to start mitosis. And then there's one last checkpoint in the middle of mitosis called the M checkpoint. And at each of these checkpoints, there's basically, this is when the cell is going to not be allowed to progress through the cycle to the next part until certain requirements have been met. And those requirements are going to ensure that things are happening the way they're supposed to inside the cell, and that cells that are supposed to be dividing are dividing correctly. And so we're going to look at each of these checkpoints and kind of the things that these checkpoints are looking for. So we'll go ahead and start with the G1 checkpoint. This is actually probably one of the most important of the 3 checkpoints, because this is at the very end of G1 before S phase. And this is basically where a cell needs to officially determine, is it going to be a dividing cell or not be a dividing cell? So a lot of cells in your guys' bodies, they don't end up dividing. They don't do mitosis. They don't produce daughter cells. We call those non-dividing cells, and those cells are usually these specialized cells in your body. And they just are doing their normal functions that are super important. And then they just sooner or later get worn out and maybe die at some point, and they don't end up dividing. And they're not actively dividing cells, constantly making more cells. And then there are cells in your body that are dividing and need to divide a lot and continuously. And so at this checkpoint, basically a cell needs to figure out what type of cell is it going to be. Is it supposed to be a non-dividing cell or a dividing cell? Because the phase after this, after the G1 checkpoint, that's when a cell is going to replicate its DNA during S phase. So S phase comes after the G1 checkpoint. After G1, you're going to now replicate all of your DNA, which is a huge task to take all of the DNA inside the cell and all of the chromosomes, which are huge, huge pieces of DNA that can be billions of nucleotides long, and you're going to copy all of it, make a second copy of all of it. And the whole point of doing that is because you are planning on dividing into 2 daughter cells who are each going to need a copy of that DNA. But if you're a cell who's not supposed to be dividing, it's a huge waste of time and energy and material to do S phase. Like, you shouldn't be replicating your DNA. So some cells will actually enter what's called a G0 phase. There are some cells who just will not make it past the G1 checkpoint because they're not supposed to be dividing cells, and they kind of take a step away from being a part of the cell cycle, and they enter this phase called the G0 phase. And the G0 phase is a state of a cell that is not going through these different phases, that's not planning on dividing. It's just a cell who's going to do what it's supposed to be doing and then eventually be damaged or broken or worn out and go away, but not divide. And so that's what the G0 phase is. And at the G1 checkpoint, that's where we'll figure out, are you a cell that's supposed to continue on in the cell cycle to S phase and mitosis? Or are you supposed to be a cell in the G0 phase? And so for the G0 phase, just to give you guys some examples, there's some cells who are permanently in the G0 phase, like they are just permanently not going to divide. They're not going to be part of this whole cycle and do all of these different phases. A good example of that are your guys' muscle cells and your nerve cells. Muscle cells and nerve cells, they are basically permanently non-dividing cells. They exist in this G0 phase and they will not do the cell cycle, which is why it's really bad if you get muscle or nerve damage, because if you damage your muscle tissue or your nerves or your brain, those cells don't really replace themselves. And so that you're permanently losing cells that aren't gonna come back. And then there's some cells who actually are in this non-dividing state, this is G0 phase, but some of them can, when needed, go back to being part of the cell cycle and start doing cell division when needed. Liver cells are a good example of that. Liver cells are usually these specialized cells that are doing important things in your guys' body, and they're not really actively dividing. They're in this G0 phase, but when needed, like let's say some of your liver gets damaged or some of the cells need to be replaced, the body can send signals to the liver cells and tell them to get out of the G0 phase and go back to being dividing cells and start replicating their DNA and go through these different phases. And so that's kind of cool. So the G0 phase, it's permanent for some cells and temporary for other cells. And then some cells, they're just never in the G0 phase. They're just always dividing. So at the G1 checkpoint, let's say you are a dividing cell. You're a cell who's supposed to be dividing. And this cell is really excited to one day divide. So it wants to start S phase. It wants to replicate its DNA. But at this checkpoint, there's a list of requirements that need to be met inside the cell before the cell is allowed to continue on to S phase, to make sure that if you are dividing, which is great if you're supposed to, that you're going to be successful in the rest of these phases. And here are the kind of things that are being checked for in the G1 checkpoint. It's kind of a long list. That's why this checkpoint is so important. Because once you start replicating your DNA, you're pretty committed to dividing. And so we want to make sure that's going to happen correctly. So one of the things that's going to be checked for is that the cell has grown to an adequate size. So the cell is not a puny little cell anymore. It's grown up. It's a big enough cell to accomplish these different phases and ultimately divide. It's also going to check that there's enough material and energy available in the cell. So if you're a cell that's like day to day barely surviving and barely getting enough energy and material to just meet the demands of a basic day-to-day functioning, then you probably aren't ready to accomplish the rest of these phases, because it's going to take a lot of material and energy to replicate your DNA and to then do mitosis and build a whole new copy of the cell. And so that's another requirement. There's also got to be adequate room in the environment for cells. And so for a lot of cells, if it's too crowded, if there's already a lot of cells and you're like all shoved right next to each other, then you probably are in a situation where no one needs more of you. Like there's plenty of cells. And so we don't need more. Whatever type of cell you are, there's apparently a lot of you. If it's crowded and there's not enough room, you probably shouldn't be dividing. It's also going to check that there's no damage to the DNA. So the cell is going to check its DNA, all of its DNA, to make sure that the DNA has not acquired some really bad mutations or really bad damage to the DNA. Because what's worse than having one set of bad DNA is replicating it and then having two sets of bad DNA that you're now going to give to future cells in the body. And so that's got to be checked and make sure that it's good to go. And then lastly, another example of something that's checked at the G1 checkpoint are the presence of growth factors. And so growth factors are these signaling molecules that are sent from other nearby cells that can bind to your receptors. And if these growth factors are present and they're binding to your receptors, that usually means that other cells want you to divide. It means like the other cells in the body are telling you, yeah, you should divide. But if there's no growth factors present, it likely might mean that no one is asking you to divide, and maybe this cell should not be dividing. So those are some of the things that are being checked for at this checkpoint. And if all of these things are being checked off this checklist, then the cell will be allowed to progress to past the G1 checkpoint to the next phase, which is going to be S phase of this cell cycle. So if the cell is big enough and there's plenty of energy and material inside the cell and there's plenty of room in the environment for the cell and the DNA is perfect and not damaged and there's growth factors present and other cells want you to divide, then yeah, you're probably good to go. You're going to move on past this checkpoint, the G1 checkpoint. And that's when you're now going to replicate your DNA. And then after that, you're going to enter G2. But then once you get through G2, you're going to get really excited. The cell is super excited to finally get ready to do mitosis and divide, which it's been super excited for this whole time. But then it meets another checkpoint. So then there's another checkpoint at the end of G2, which if I go back to this picture here, at the end of G2, there's another checkpoint here that's going to be making sure that you're ready to move on to mitosis, that you're ready to finish this up correctly, this whole cell division thing and this cell cycle. And so in the G2 checkpoint, there's a couple big things that are going to be checked for before the cell's allowed to move on to the next part, the next major set of events, which is going to be like prophase, metaphase, anaphase, telophase, and cytokinesis. At the G2 checkpoint, it's going to check, first of all, that, again, there's enough material and energy available in the cell. So if you barely survived replicating your DNA and you're now a cell that's starving and not having enough material or nutrients or ATP that's being made in the cell, then you probably aren't ready for the huge task of mitosis, which is an incredible, it's like the biggest task that a cell can accomplish, which is dividing. And so that's one thing that's checked at the G2 checkpoint. Another thing that's checked at this checkpoint is that again, the DNA is going to be checked to make sure that the DNA is not damaged, specifically the newly replicated DNA. So you just finished S phase where you took all of the DNA and you made a second copy of all of your chromosomes. You duplicated all of your chromosomes, you copied all of the DNA. And that often can result in some messed up DNA when you're doing DNA replication. DNA replication is actually the number one source of mutations on this planet, is that when cells are copying their DNA to do cell division, they mess up. There's a lot of nucleotides you're putting together. There's A's and T's and G's and C's in a specific order, and there's billions of them that you have to build to copy all of the DNA and all the genes and instructions needed for the cell. And at the G2 checkpoint, it's going to make sure, did you replicate the DNA good enough? Did you do a good job, and did you mess up? And if you messed up, then you're not going to be allowed to go past this checkpoint, because you don't want to give that bad, messed up DNA that you made to a daughter cell, because then you're just making the problem worse. And if cells who do have DNA damage, who did mess up during DNA replication, if they're not able to fix that or repair that, then those cells will undergo something called apoptosis. Apoptosis is when a cell is programmed to kill itself, basically, which is actually a really good thing. Like if you are a cell with bad DNA or something's going wrong during the cell cycle and you're not making it past these checkpoints, then apoptosis is probably the best thing that could happen, because you don't want that cell to be dividing. You don't want that cell with bad DNA, or that cell that's messed up, or that cell that's not meeting these requirements. You don't want that type of cell to divide and make bad daughter cells. You want that cell to basically just give up. And then now that cell is just going to program itself to die. So that way other cells, good cells, can replicate instead. So instead of having really, really bad, cruddy skin cells with bad DNA dividing, making more bad skin cells, how about you kill yourself, these bad cells with their bad DNA, and undergo apoptosis, and let the good skin cells replace themselves or make more copies of themselves. This is good. So apoptosis is not a bad thing. This is a good thing. This is what is supposed to be happening in a normal, healthy organism, is that the bad cells should not be allowed to divide. Okay, you need to stop them at all cost, which apoptosis is a great way to do that. So anyway, let's say you made it past the G2 checkpoint. You have plenty of energy and material to move on to mitosis, and your DNA that you replicated looks great. And so now you're going to move on to mitosis. You made it past the G2 checkpoint here. And now you can start prophase and tearing down the nucleus and condensing your chromosomes. And then those chromosomes are going to start lining up in the middle during metaphase. But then you hit this last checkpoint where the cell is not going to be allowed to finish off the cell cycle with mitosis and cytokinesis, the rest of mitosis and cytokinesis, until this last checkpoint is successfully passed. And at this last checkpoint, it's actually at the during metaphase before anaphase that you hit this last checkpoint. So this is going to be right before anaphase. You're going to see this checkpoint take place. So during prophase or during metaphase before anaphase, there's one last checkpoint. And what's going to happen is there's one really important thing that the cell needs to check before moving on to anaphase. To make sure that all of this wasn't a waste and we're not going to mess up here at the end, because we're really close now to finishing mitosis and producing 2 daughter cells. And if you guys remember in metaphase, that's when the chromosomes are lining up and these microtubules are supposed to be attaching to the kinetochores on each sister chromatid. So these duplicated chromosomes are made up of 2 sister chromatids, 2 copies of each other. And during metaphase, those are supposed to be all latching on to these microtubules on both sides, attached to the sister chromatid, sister chromatid, sister chromatid, sister chromatid on both sides. And then they're going to be pulled apart during anaphase. But before you start anaphase, at this last checkpoint, the cell needs to make sure that every sister chromatid is attached to these microtubules. So these microtubules should be attached to every single kinetochore on each of these sister chromatids, because you're about to pull them apart. And if you end up pulling them apart before all these microtubules have a chance to successfully attach to these sister chromatids, you're going to end up with a situation like down here. Like, let's say that not all of them attached. Let's say this microtubule right here did not attach to this sister chromatid. We call which is not good, because let's say you start anaphase, then what's going to happen is these microtubules are going to shorten and pull apart these sister chromatids. But these guys, if this guy wasn't latched on, they're going to be pulled together towards this end of the cell. And you're going to end up with these chromosomes here packaged together in a daughter cell. And that daughter cell is going to end up with an extra copy of a chromosome that it's not supposed to have. And on this daughter cell, the one that's forming on the right side, that's going to form after cytokinesis, this daughter cell on the right side is missing a whole copy of a chromosome with thousands of genes and important instructions on it. And so at the very end, you actually made a huge mistake where you did not successfully divide the chromosomes between the 2 daughter cells that are forming. And so that's called non-disjunction when that takes place, and cells will usually be really good at avoiding that because of this M checkpoint, which will only let a cell start anaphase once it's made sure that these microtubules have attached. So that's why that last checkpoint is there and why it's so important, because that would suck to produce these terrible daughter cells that are genetically messed up after doing all this work for nothing, basically. Those daughter cells are not going to function properly. So those are the checkpoints, and that's what they're checking for. So it's a good way to regulate and make sure things are happening correctly inside of a cell that's trying to divide. Now, to continue in more detail about how this is all taking place on a molecular level, it gets kind of confusing. And so we're going to go through something that's going to seem a little tricky at first, and I'll try to make my best to make it make sense. But these checkpoints are ultimately being controlled by these regulatory proteins that are inside the cell. So there's these proteins inside the cell who are ultimately controlling whether or not a cell is allowed to start the next part, if it's allowed to continue on past these checkpoints to the next phase of the cell cycle. And the 2 type of proteins that are controlling this are called cyclins and CDKs. So CDK stands for cyclin-dependent kinases. So kinases, this is a type of protein that we learned about in the 4.1 notes, actually, during signal transduction pathways. Kinases are proteins who phosphorylate other proteins. So there's these kinases involved in the cell cycle, and these kinases, these are proteins, their job is to go phosphorylate other proteins. And by phosphorylating other proteins in the cell, those proteins become active when they get phosphorylated, and then they start doing things in the cell that weren't happening moments ago. So these CDKs are basically waking up certain proteins inside the cell, telling them, okay, you go start doing your job, you go start doing your job, you go start doing your job. And he's doing that by phosphorylating them. But these kinases, we say that they're cyclin dependent because these kinase proteins are only active when they're bound to a cyclin molecule. So cyclin is a protein in the cell that binds to CDKs, it binds to cyclin-dependent kinases. And when cyclin and CDKs bind together, they form this complex that's now active. Now this CDK is able to go phosphorylate other proteins, which is what you guys see happening in this picture. So here's a protein that this CDK is now going to go phosphorylate. And he's only able to do that when he's bound to cyclin. So if there's no cyclin, he's doing nothing inside the cell. But when there is cyclin, he can bind to cyclin and start phosphorylating other proteins inside the cell. And this is what ultimately is going to allow a cell to move past these checkpoints. If these CDKs are binding to cyclin and becoming active, that's a sign that a cell is now turning on certain proteins that are needed for the next part of the cell cycle. Like, for example, when a cell is going to go into S phase, like if you get past the G1 checkpoint to start S phase, the only way you're going to start S phase is by turning on the proteins needed to replicate your DNA. So during S phase, that's when the DNA and the chromosomes are going to be duplicated. Well, that doesn't just happen magically. There's proteins who need to turn on and be phosphorylated to start taking all of your DNA and start building new DNA, start building and synthesizing a copy of all of that DNA. And these CDKs, it's their job to go do that. They need to go find the proteins needed for the S phase to do DNA replication, to go phosphorylate those proteins to get that stuff started. And if these CDKs aren't active, then that's not going to happen. You're basically stuck at the G1 checkpoint. You're not moving past G1 until these CDKs go and start phosphorylating these guys. Same thing with like at the G2 checkpoint. At the G2 checkpoint, you want to start mitosis. You want to start prophase next. Prophase is after G2. And that's when you're going to start condensing the chromosomes and you're going to start building these microtubule protein filaments that are going to start attaching to the chromosomes. You're going to tear down the nucleus. And all of those things don't just happen magically. There's proteins who now need to become active and start doing that stuff. And again, there's CDKs whose job is to go turn on the proteins needed to start prophase and to do mitosis. So these guys ultimately control a cell's progression through the cell cycle. So just vaguely, if anything, just vaguely understand that these CDKs, these cyclin-dependent kinases, which have to bind to cyclin proteins, they're the ones making a cell progress and go to the next part of the cell cycle. And if these guys are not active in the cell, then you're probably not, the cell's stuck. It's not moving on to the next set of things that needs to happen. And so the way this kind of works at a checkpoint is basically these CDKs are always present. These cyclin-dependent kinases are always present, but they could be like asleep and inactive, doing nothing. And they're not active until cyclin comes along. But cyclin is this protein that will accumulate in a cell, but then quickly be degraded, and then it goes away. So cyclins come, but then they go away. And when they're present and they start accumulating in the cell in high concentrations, they start to bind to these CDKs, and then things start happening when these cyclins are present. But when the concentration of these cyclins go down, these CDKs go back to doing nothing. And so what you're going to see happening inside the cell is you're going to see the concentration of these cyclins fluctuate. At certain parts of the cell cycle, the cyclin concentration is going to go up. And then that's going to start activating these CDKs. You can see the activity of these CDKs who are just floating around in the cell. They're going to start to become active because they're going to start binding to the cyclin. And then they're going to go start doing things to move the cell on through the cell cycle. And then quickly, though, those cyclins, if you guys look at this graph here, those cyclins, then the concentration crashes because those cyclins quickly get destroyed. And then those CDKs go back to sleep. They go back to being inactive. And then they stop activating the proteins that they were activating. And then it repeats the next time around in the cell cycle. When the cell goes through the cell cycle again, those cyclins start to accumulate, activate these CDKs, and then those CDKs become active, and then the cyclin goes away, and then those CDKs stop doing whatever it was they're doing. And so that's the gist of how this works. That's also why we call them cyclins. We call them cyclins because these are proteins whose concentrations cycle throughout the cell cycle. Like they increase and decrease at regular intervals during the cell cycle, causing these CDKs to become active at predictable times during the cell cycle. Now, what makes this a little bit complicated is that there's different cyclins inside the cell who can activate different CDKs. So there's not just one type of CDK in the cell. There's different CDKs, cyclin-dependent kinases, who become active at different times in the cell cycle. So like there's CDKs who are waiting for certain cyclins to come, that become active, and they go turn on certain proteins needed to move the cell to the next phase, the next part of the cell cycle, and then they go away. And then now the cell is in the next phase of the cell cycle, like it's going through S phase maybe and replicating its DNA. And then that stops when it's done. And then there's another type of cyclin that starts accumulating, and that cyclin binds to a different set of CDKs, another type of CDK proteins, and then those CDK proteins become active, and then they go turn on a different set of proteins. Maybe the proteins needed to start prophase and start mitosis. And then quickly those cyclins go away and those CDKs go back to sleep. And then there's going to be another set of cyclins, a different set of cyclins that start to accumulate later on in mitosis. And they're the ones that activate another set of CDKs. And those CDKs turn on proteins and phosphorylate proteins that are needed to finish up mitosis and do cytokinesis. And so it's these different cyclins accumulating, activating certain CDKs that allows a cell to move on throughout the cell cycle, and then they go away, and then new cyclins come and turn on a different set of CDKs, and those CDKs go and tell proteins what to do, and then those go away, and those CDKs become inactive again, and then there's another type of cyclin that starts to accumulate in the cell, and that turns on a different group of CDKs who then tell the cell what to do, and then they go away, and so then you're going to have this overall during the cell cycle, you're going to have these different CDKs becoming active in regular intervals, turning on different sets of proteins, phosphorylating different set of proteins, which is ultimately going to allow these phases to actually take place, to allow these new events to take place inside the cell so that the cell can do these different things that are part of these phases in the cell cycle. So I hope that vaguely makes sense. It's ultimately comes down to these cyclins working together with their CDK buddies to control the progression through the cycle. And if these cyclins aren't present, these CDKs aren't doing anything. And so a cell that's being stuck at a checkpoint, like let's say you're stuck at the G1 checkpoint, it's probably on a molecular level, what's happening is the cyclin needed to go past the G1 checkpoint, it's not there. So maybe like in this picture, if you look at cyclin E, to get past the G1 checkpoint, to go to S phase, you're supposed to have a lot of cyclin E inside the cell, which is going to turn on the CDKs needed. But if you're a cell that's not progressing past G1, there's probably something going on that's wrong. Maybe your DNA is damaged. Maybe there's not enough room in the environment. And that cyclin won't be present. There's going to be low levels of that cyclin in the cell so that the cell can't get past that checkpoint. It can't turn on the CDKs needed to then allow the cell to go through to the next phase. So that's this, well, in a specific way, kind of detailed way, that's kind of what's going on with these cyclins and these CDKs. And so hopefully that kind of makes sense. Now, there's a few more things in these notes. I know it's kind of long. There's a lot of, this is a big topic here, topic 2. Take a break if you need to. But I want to give you guys some examples of how at these checkpoints a cell might be stimulated, which means like it's being encouraged to get past the checkpoint, or how it might be inhibited, meaning like the cell, something's happening in the cell that's trying to block it from getting past the checkpoint. So for the G1 checkpoint, here's how a cell might be stimulated. Here's a specific example of a cell being stimulated at the G1 checkpoint, meaning that the cell is being encouraged to like, yeah, you should make it past the G1 checkpoint. At the G1 checkpoint, the presence of growth factors, if you guys remember me telling you that is one of the requirements to get past the G1 checkpoint. And there's a specific type of growth factor called platelet-derived growth factor that actually will bind to special cells called fibroblast cells and cause those cells to get past the G1 checkpoint. So this PDGF, this platelet-derived growth factor, it actually, when your guys's, when your blood arteries are cut or injured, your blood vessels are injured, these PDGFs will be released. By injured cells, these growth factors, and they're going to bind to fibroblast cells. And fibroblast cells are these cells in your body that help to clot your blood, and they help to heal wounds. And so what happens is those cells, when they bind to the PDGF, it actually allows the G1 cyclin CDK complex to become activated. So that first set of CDKs with the first set of cyclins is now going to start becoming activated once this guy is binding to the receptors on those special cells. And so this is encouraging the cell to then move past the G1 checkpoint. Anytime you're getting these cyclins and these CDKs to become active, that's going to help get a cell to move on to the next phase of the cell cycle, to get past the checkpoint. And these PDGFs, that's what they're causing to happen. They're helping to activate the cyclin CDKs needed to get past G1. And so that's a specific example of how a certain type of cell is stimulated at the G1 checkpoint. Here's an example of how it might be inhibited. Like I said, if it's too crowded, cells will stop dividing. So there's certain types of cells that when there's not a lot of cells next to them, they will be actively dividing. But then once it becomes crowded, they stop dividing. And the reason they stop dividing is once it becomes too crowded, there's these signals that get activated when it's too crowded, when you're shoved right next to other cells. There's these signals that get initiated inside the cell that actually inhibit the CDK cyclin complex needed to get past G1. So it starts inhibiting the cyclins and the CDKs needed to move on past G1. So now you're going to be stuck because those CDKs and cyclins, those are what get you past the checkpoint, and now they're being inhibited because of the density of cells being too high. We call that density-dependent inhibition, which for a lot of cells, they have density-dependent inhibition. When there's too much cell density, they stop dividing, which is actually a good thing. Like, you don't want those cells to divide. And then if, let's say, you took some of those cells out and now those signals go away, those crowded signals go away, well, then these cells will start no longer inhibiting those CDK cyclin complexes, and then now the cells will start making it past the G1 checkpoint and continuing on in the cell cycle and dividing more. And then at the G2 checkpoint, an example of stimulation at the G2 checkpoint is the accumulation of these mitotic cyclins. So there's these cyclins involved in starting mitosis. This is a specific type of cyclin. Specific type of cyclins needed to get a cell past G2. And when things are going correctly, when things are occurring correctly in the cell, those cyclins start to accumulate more and more and more. You start to have more and more of these cyclins in the cell, and they bind to the special CDK towards the end of G2 that form this activated CDK complex called MPF. MPF is a mitosis promoting factor, but it's basically just an activated CDK. It's a CDK, a special type of CDK that came together with a special type of cyclin, and this is the CDK and cyclin that's going to start mitosis. It's going to start turning on the proteins and phosphorylating the proteins needed to get mitosis to start. And so it has a special name. It's called the mitosis promoting factor, which is what's going to allow the cell to now get past G2 and this checkpoint to start mitosis. But that's only going to happen if things are happening correctly inside the cell. If your DNA was replicated correctly and you have plenty of energy and resources and material inside the cell, this cyclin will start to accumulate as a response to that, which will get you past this checkpoint. Whereas, let's say there's a cell that needs to be stopped at the G2 checkpoint. There's a protein called P53. It's a really important protein, comes up in lots of examples. P53 is the special protein that actually checks your guys' DNA, and it checks for damage to the DNA. And when it finds damage to the DNA, it actually inhibits the cell from moving on in the cell cycle. And so if there is damage that it finds in the DNA, it'll actually go ahead and cause the CDK and cyclin to be inhibited. It'll find the CDKs and the cyclins and inhibit them from progressing the cell through the cell cycle, which is a good thing. Like if this guy finds damage in your DNA, he needs to go stop the cyclins and CDKs from turning on proteins to progress you through the cell cycle. So that's an example of inhibition. This guy checking your DNA and finding a mistake, and then going and inhibiting, causing the inhibition of these CDKs needed to move you on past the checkpoint. And P53, he's actually involved in the G2 checkpoint. After your DNA is being replicated, he checks the newly replicated DNA. He's also involved in the G1 checkpoint. He does check, at the G1 checkpoint, he also checks the DNA before it's replicated. And again, if in either case, before or after it's replicated, it finds mistakes, it's going to inhibit the CDKs and the cyclins from becoming activated so that the cell can't move on to the next phase. And so ultimately, cells will do one of three things, right? So like in terms of like what's going to happen to a cell during its life, there's three main outcomes. Option one is you're just a cell who's going to undergo mitosis and you're going to divide. And that's usually true of cells who are exposed to a lot of growth factors. And these are cells that are supposed to be dividing, like certain types of stem cells that you guys have that need to make new cells. They're actively dividing, or like your skin cells, they're actively dividing, making more cells. And so that's option one. Some cells, they just like to end up dividing, and then they divide more and more and more. Option two is some cells, once they form, they actually turn into specialized cells, and they turn into certain types of cells, like blood cells or muscle cells or heart cells or liver cells. And we call that cell differentiation when they become specialized cells. And those specialized cells, some of them usually stop dividing. They won't do mitosis. And so, like I said, those are non-dividing cells that will just eventually die off at some point. But for a long time, they can just live and do their job, and then maybe they'll get damaged at some point and no longer work, but they can last a while. And so that's option two. Maybe you're a cell that's not supposed to be dividing. And then option three, there's some cells who are supposed to just kill themselves, go through apoptosis. So sometimes there's certain cells that your body needs, and then at some point it no longer needs those cells, and it tells those cells to go away. Don't divide, but don't stick around. Just go through apoptosis, and then those cells go away. The common example of that is during development. During development, when you guys are developing, especially as an embryo, into a baby, a human being looking thing, there's different stages of development where at some point there's certain cells that need to be there to start building the organism, but then those cells are no longer needed later on in development. So a good example is like your guys' fingers and your toes. They used to have skin between them as they were forming. And there were skin cells there. You had webbed feet and webbed fingers. But then later on in development, those cells between your digits are told to go through apoptosis, and then they go away, and you're supposed to now have these free moving phalanges in your toes and in your fingers. And so that's another option. So either a cell is going to be dividing, or a cell is going to be alive and not dividing, or a cell is going to go away and go through apoptosis. And then the very last thing in these notes is, since we're talking about cell cycle and the regulation of the cell cycle, we have to talk about cancer, because cancer is ultimately caused by uncontrolled cell division. It's when the cell cycle is not being regulated correctly and cells are dividing in an uncontrolled and unregulated way, ultimately producing bad daughter cells, and then those cells produce more bad daughter cells, and then you keep doing that and you end up forming tumors that are not good to have in your body. And so all of this is what can explain how cancer forms. It's by this stuff that everything that I talked about in this video. If that stuff's not happening correctly, one of the common outcomes is cancer. If cells are not dividing correctly, or cells that are not supposed to be dividing, are the ones that are dividing incorrectly, making bad daughter cells, and they're not regulating themselves at these checkpoints and making sure things are happening correctly, that's how you end up with tumor formation and ultimately cancer. And so there's two types of genes that are kind of involved in this whole thing with the cell cycle being controlled. There's things called proto-oncogenes and tumor suppressor genes. And these are two categories of genes that code for a huge variety of proteins. So genes code for proteins, and these proteins have specific jobs. And there's a certain category, a broad category of proteins that are coded by, that have one thing in common. Like they might be doing a variety of things, but there's a certain category of proteins called proto-oncogenes, which I guess are the genes that code for these proteins. And these proteins, the one thing they have in common, they're doing a lot of different things inside the cell, but one thing that they have in common is that they're promoting the cell cycle in certain ways. And so if you have a protein, and that protein's job in some way is promoting and encouraging cells to get past these checkpoints and to divide, we would call that a proto-oncogene. Okay, that's a product of a proto-oncogene. That's a protein who's helping cells progress through the cell cycle, which is good. You need cells to divide, and you need proteins to do certain jobs to encourage cells to divide. And then there's a class of genes, a category of genes called tumor suppressor genes, which make a variety of proteins that do a variety of different jobs, but they have one common underlying one underlying thing in common, and that is in some way their job is contributing to the cell cycle being inhibited and preventing cells from continuing on in the cell cycle. So these are proteins whose job in some way is telling cells don't divide, or it tells cells, these are sometimes proteins that lead to apoptosis, that tell cells actually definitely don't divide, you need to kill yourself. And so proteins that are involved in halting or stopping or slowing down the cell cycle or slowing down cell division or leading to apoptosis, we call those tumor suppressor genes, which are actually also a good thing. There are cells in your body that aren't supposed to be dividing. You should have these proteins in place telling those cells, no, don't divide, that are slowing down and inhibiting them from progressing through the cell cycle, or ultimately telling them to do apoptosis when those cells are bad or need to go away. Like, this is a good thing, actually. So both these types of genes make proteins that are very important. Proto-oncogenes make good proteins that help cells divide. Tumor suppressor genes make proteins that slow down cell division and make cells not divide. Both of them are needed to have controlled cell cycle regulation. But the problem is, if you get mutations in these genes, it could lead to their proteins not working correctly. And depending on what protein it is, that could cause the cell cycle to then start getting uncontrolled when these things aren't working anymore. These things that are supposed to be accelerating are supposed to be halting the cell cycle. If those things aren't working anymore, well, now you're going to have uncontrolled cell division, which can ultimately lead to cancer. And so we kind of think of it, well, this picture gives you a little analogy. Think of the proto-oncogenes as the gas in a car, and the tumor suppressor genes as the brake. And so tumor suppressor genes slow down the cell cycle and slow down cell division. Proto-oncogenes increase cell division. And in cells, you need both. You need the brakes and you need the gas. You need cell division to happen faster, and then there's times where you need it to happen slower. And you need both of those for a normal, healthy, functioning cell. But if you mess those up, if you have mutations in the gas, for example, let's say your gas is way too sensitive. Like now, all of a sudden, like the car, you're barely pressing on the gas pedal, and your car goes from 0 to 100 immediately because your gas pedal is broken. Something's not working correctly. Well, that can lead to cells dividing uncontrollably. Or let's say the brakes aren't working. The brake pedal, you try to push it and you shove your foot on it and it's not stopping, and cells are just dividing way too fast because the brakes aren't working, the tumor suppressor genes. That could also lead to uncontrolled cell division. And so to give you guys some examples of these type of mutations that could lead to cancer, if you have a mutation in a proto-oncogene, that could sometimes lead them to become oncogenes. Oncogenes are bad proto-oncogenes. These are proto-oncogenes that are now that have mutations that are overstimulating the cell cycle. Okay, so now these proteins, they're basically they're like proteins who are too good at their job. So these proteins have accumulated a mutation that make them way too sensitive or make them way too effective at doing what they're supposed to be doing, which, if you remember, the job of a proto-oncogene is to increase cell division. And sometimes you could lead to a mutation in these proteins that make them increase cell division too much, like way more than they should be, way more than they're normally supposed to. And that could lead to increased cell division and ultimately tumor formation and cancer. So for example, if you have a mutation in a gene that codes for CDK, that makes it hyperactive. Hyperactive means more active than usual. So you have the CDK. CDK is what controls whether or not a cell goes through the cell cycle. And this CDK is hyperactive. Maybe the CDK is like phosphorylating other proteins, even when there's no cyclin. Like CDK is only supposed to be active when there's cyclin, but let's say there's no cyclin and this CDK is broken and it's still phosphorylating other proteins. It's like it's doing its job. It's more active than it's supposed to be. Well, then you're going to have faster progression than the cell cycle than they're supposed to be. That's bad. You don't want cells to be going through the cell cycle and dividing more than they're supposed to be normally. And if these CDKs become more active than they're supposed to be because of a mutation, that could lead to increased cell division, and that's what causes cancer. So that's one example. And so a CDK, you could call a CDK protein a proto-oncogene. It's a normal protein that's supposed to help cells progress through the cell cycle. We would call it a proto-oncogene. It's kind of like the gas, but if that gas is broken in a way that becomes overstimulated or hyperactive, it's not good. That's when you end up with an oncogene, which is a bad proto-oncogene. An oncogene is a cancer-causing protein. And then one more example with a tumor suppressor gene. So tumor suppressor genes, again, those are proteins. Those are making proteins that are supposed to be inhibiting the cell cycle and slowing down the cell cycle. But sometimes you might have a mutation in a tumor suppressor gene that leads to the protein. Let's say the protein is non-functional, so the opposite of hyperactive. Like this protein is now underactive. Like this protein maybe doesn't work anymore. Like there's a mutation that causes this protein to not fold correctly, and now it can't do its job at all. And its job was to slow down the cell cycle when needed. Well, now if you have this protein who can no longer slow down the cell cycle when needed, you're going to end up with cells dividing and going through the cell cycle more often than they're supposed to be. And that, again, can lead to tumor formation with increased cell division and cancer. And so a specific example is like P53. P53, I showed you earlier, that's the protein that checks your guys' DNA for damage. And when it finds damage to your DNA, it tells the cell cycle to stop. Like it tells cells, do not continue on in the cell cycle, do not divide, do not do mitosis, which is good. Like that's a good job to have. But let's say you have a mutation where P53 is inactive, like he's not able to do his job. So there's damage to your DNA, but P53 is not working. And so no one's telling the cell to stop, and the cell continues to divide anyway, producing these daughter cells with bad DNA. So now you have a situation where cells are dividing when they're not supposed to be dividing, which is the cause of cancer, okay? So anyway, I hope that kind of makes sense, the role of these tumor suppressor genes and proto-oncogenes in maintaining regulated cell division, and how when you mess those up, that could lead to uncontrolled cell division and possibly cancer. And that's it for these notes. I know this was a super long video, you guys, but I hope that makes a lot more sense now. A lot of complicated things in these notes, but that's the last notes of Unit 4 in the last video. So anyway, thank you guys. I'll see you later.